In standard biology, DNA is treated as a highly stable, macroscopic blueprint. It is a structure designed to be resistant to errors. The double helix holds our genetic letters firmly together, locking them into a specific, predictable order. That classical timeline explains how physical traits accumulate in a species, but it doesn't account for the microscopic origin of the mutations themselves. To find the origin, we must zoom inside the double helix. These are the genetic letters, A, T, C, and G, held together by hydrogen bonds. The actual physical glue bridging these base pairs together is incredibly light. It consists of single protons. A genetic mismatch occurs when a proton ends up on the wrong side of that bond. This chart shows the potential energy curve between the two sides of that bond. Notice the massive spike in the middle. In classical physics, a single proton does not have the energy to climb over that barrier. We are left with a physical contradiction. We see genetic spelling mistakes that, by the laws of classical physics, don't have the energy to occur. The answer lies at the subatomic scale, where protons stop acting like solid objects and behave like spread-out waves. Because of this wave nature, the proton doesn't have to climb the energy barrier. It simply teleports straight through it, a phenomenon known as quantum tunneling. This is the standard, stable form of a guanine-cytosine base pair right before a jump occurs. Here is the exact midpoint of the reaction, as the proton actively tunnels across the hydrogen bond, and this is the resulting mutated structure. The proton is now on the wrong side, creating a rare, altered arrangement known as a tautomer. These subatomic teleportations happen at quantum speeds, rewriting a genetic letter in a fraction of a second before the rest of the cell can respond. There is a catch. Quantum tautomers are highly unstable. Left alone, the proton will almost instantly tunnel back to its correct position. But whenever a living cell copies itself, a biological machine has to physically unzip the entire DNA double helix. Recent research reveals that this unzipping enzyme, called helicase, operates about 100 times faster than scientists previously predicted. This creates a microscopic speed trap. Helicase rips the strands apart in the exact microsecond the proton is on the wrong side. That mechanical separation stabilizes the tautomer. The strands are pulled apart, locking the proton on the wrong side so it cannot jump back. Helicase acts as an ultra-fast biological camera shutter, permanently capturing a fleeting quantum fluctuation before it can reset. Now separated, that tautomeric strand enters the cell's standard replication machinery to be copied. Because the proton is trapped out of place, the copying machinery misreads the letter. It assigns a completely wrong partner base to the new strand. This generates a brand new DNA double helix containing a permanent point mutation, which will be passed on to the next generation. Through this rapid sequence of events, a transient quantum ghost is hard-coded into a biological reality. This research proves quantum mechanics isn't restricted to absolute zero temperatures or laboratory supercomputers. It is actively operating inside our warm, wet cells right now. This microscopic tunneling drives massive macroscopic consequences. It is an invisible engine pushing evolution, biological diversity, and diseases like cancer. Life itself relies on the bizarre rules of the quantum realm, entirely shattering our illusion of a slow biological clock.